Hello everyone, I'm Srikrishna Narasimhan from Museum Institute of Zoology, Polish Academy of Science. I'm going to talk about the ongoing PhD project of mine on the title Are Invaders Smarter? Individual Cognition versus Social Information in Invasive and Native Ants. What are these native ants? Is that they're Lepidoptera humile, which are native to South America, especially Brazil and Argentina, where they form large colonies and are polygynous, which is that they have hundreds of queens and thousands of workers. And because of human intervention, they were transported to different parts of the world, including North America, Europe, Africa, Australia, and Japan, and many other places. So when they invaded Europe, they started forming vast super colonies, even with the new habitat changes that had occurred between them. And they are among the 100 worst invasive species in the world. And we are going to compare how they were able to be such a successful invasive species by comparing them to a native species, which is Tapinoma erraticum, which is also polygynous and is found in Europe. Uh, as a native species, and how they do problem solving in these new arenas by comparing them with uh, problem solving in the native area. What we have hypothesized is that when these invasive species depend more on social information rather than on individual cognition, because they cannot spend enough resources to develop proper brains for them. So generally problem solving in ants can be because of two different methods. One is social information and the other is behavioral plasticity, which encompasses inter-individual plasticity and intra-individual plasticity. So let's go on what is a social information or social cues. So ants generally, when they find some food source or a danger, they drop pheromones on the trail for other ants to follow, and this is called trail pheromones. And these help the other ants in navigating and solving problems in their area. So we want to test how this affects the behavior of the ants. Since we cannot force the ants to drop pheromones where we want, we used a proxy of Z9-hexadecanol, chemical compound which is found in the gaster in trace quantities in the gaster of Lepidoptera humile, and also is widely used as a trail disrupting technique in different parts of the world to help in solving the invasive problem of Lepidoptera humile. Also, these attract Tapinoma erraticum. So, and then next is what is behavioral plasticity. So, behavioral plasticity is when an organism changes its behavior according to different environmental conditions based on the habitat. So when a behavioral trait does not change across observation, we can say that there is no plasticity. What if there's a constant increase in the behavioral trait across observations, but it is uniform in different species of the ants, because ants are not solitary, they are social species. This means that they have intra-individual plasticity, is that each ant is able to change behavioral trait in the same manner as other ants of the species. And if the behavioral trait changes randomly in different ants, then it means that they have inter-individual plasticity, that the different ants have different ways of behaving. So we're going to test how these fare against Lepidoptera and Tapinoma. So we collected the ants from Ria de Arousa in northwest Spain, in Galicia, in different locations. The red dots on the map that you see are of Lepidoptera collection, and here there is a red dot hidden in Refourches, because we found 3 or 4 Refourches colonies there. So we have found Tapinoma colonies there, and the blue ones are Tapinoma erraticum colonies. And as you can see, they share similar kind of ecosystems where they can occur. So what we did with them is that we used callow workers, and we tested them after 10 days of their emergence on 5 days on these 3 behavioral tests. The first behavioral test was exploration, the second neophobia, and the third maze solving, consequently one after the other for 5 days. So in exploration, they were allowed to explore an empty arena with 4 walls but an open center. In neophobia, the open center, we added a neophobic new element for the ant. So neophobia means fear of new items. So we assume the pyramid shape that we added was something which they do not see in nature, and the color blue is that they rarely see in their surroundings in the natural habitats. Next, we saw how they solved mazes, where we added rewards on one of the arms for 5 different days, and we checked how they were able to remember or solve the maze to reach the reward. And we had 2 different treatments for them. One was with social information, is that where we had pheromones as we discussed earlier, and the other without any social information. And also we had controls where we let the ant explore an empty arena without any walls, which was so that to check how the brain changes without these behavioral tests, and how the brain changes with these behavioral tests. So for controls to not have the effect of the fluid of the pheromone, we added ethanol in other remaining locations like this, where for the exploration, one sector we added the pheromone close to the wall, and the others we added ethanol. In neophobia, the one with the social treatment was sprayed with pheromones, and with the one without the social treatment was sprayed with ethanol. And the same with maze, where the arms leading to the reward was added with a drop of pheromone, whereas the others they were added with ethanol. And these are an example of how they were exploring the 3 different behavioral tests in exploration, euphoria, and maze solving. And after the 5 days, their head was cut off and the brain was extracted to check neural plasticity. In neural plasticity, we basically check 2 different things. The first is microglomeruli density, which is microglomeruli means microstructures of synaptic complexes which are present in the mushroom bodies of the ants in the brain, and these are responsible for learning and memory. So we assumed with the 5 behavioral tests that we performed that these would have changed a bit, and to check how these were changing. So the horseshoe shaped structures that you see on the top of the brains in the left GIF are these mushroom bodies that we checked. And next we also checked how the volume of the brain was changing based on the behavioral test that we performed. We measured the volumes of the brain, and this is still a work in progress. We're still working on it, so I do not have any more information or nice pictures to show you based on this. So let's jump to the preliminary results that we got from these behavioral tests. We saw that the exploration decreased through observations across the 5 observations for all the, including all the species. This does not tell a lot about them. And we saw that Tapinoma focalans explored more than Linepithema in exploration, but the exploration control was much higher. But this can be associated with other reasons which we will discuss later. And next, for the neophobia test, we first checked how they use the use of the neophobic item, the proportion of time they spent on the neophobic item, and we found that there was no difference among the species, but we see that they were attracted to the neophobic element across different tests, which is quite intuitive. And the next, in maze solving, we saw the latency to reach the reward, uh, and we found that there was no difference between the species or treatments. So these results are quite contrary to what we expected, because we expected Tapinoma ants to explore less with comparison to Linepithema with the social information, but this was not the case. And we also found that the pheromones that we added did not have much effect on the treatments, but it is still worthwhile to check deeper on how this affects the plasticity in behavior and the plasticity in the brain. Like for example, the graph that I showed is it's still a work in progress, is how the plasticity of the explorative behavior changes across 5 different observations in Linepithema and Tapinoma. And these are calculated using GLMMs, and this can show a wide can lead us to some interesting conclusions. So as we are still working on it, I'm a student. Any suggestions or discussions are always welcome. So finally, I would like to thank the lab members of Social and Myrmecophilous Insects in Museum Institute of Zoology for helping us and motivating us through the experiment and solving the doubts, and the brain team, which are very busy in analyzing the brains. So the person is Animalta Software. You can follow us on our lab activities on X, or also the progress of the project in Alien Rules. And finally, I would like to thank Argentina ants and Tapinoma ants for cooperating with the experiment and sacrificing their life and brains for it. So thank you.